Okay. My name is Hector Bravo. I am a professor of engineering here in the College of Engineering and Applied Science, and it's my pleasure to introduce today Dr. Marcia Silva. This is the bio that Dr. Silva prepared. She is an assistant scientist and the facility manager for the UW-M Water Technology Accelerator located at the Global Water Center. She earned both her master's and Ph.D. degrees in engineering from UW-M. She has more than 20 years of industrial and academic experience, has authored several journal papers and book chapters, and has presented her work at national and international conferences. After earning her doctorate in environmental engineering from the College of Engineering and Applied Science in 2013, by the way, I was her co-advisor, Dr. Silva took on the huge task of creating the water research facility associated with the rise of the Global Water Center in Milwaukee. In 2017, she received the UW-M Graduate of the Last Decade Award as a recognition for being instrumental in the success of the research arm of the Global Water Center and has impacted the research reputations of UW-M School of Freshwater Science and the College of Engineering and Applied Science. Her lab just received national recognition for the development of a multi-purpose water sensor, reaching the top 10 finalists of NASA iTech competition. For the past 5 years, she has been working collaboratively with industry, startups, water council, and government organizations in the freshwater space, focusing mainly on development of novel water sensors and novel filtration media for water purification. Dr. Silva serves as an adjunct faculty member at the UW-M School of Freshwater Sciences, Material Science and Engineering, Industrial and Manufacturing Engineering, and Electrical Engineering and Computer Sciences, and is an active member of the Milwaukee Water Hub. She has mentored over 60 students to date, including postdocs, graduate students, undergraduate students, and high school students considering science as a career. So let's welcome Dr. Silva.
Thank you so much. Thank you so much. Such a pleasure to be here today. So when I was preparing these slides, Dr. Marcia invited me to be here today, and I was very pleased with her invite. I was just thinking, how can I tell my audience in just the few minutes that we have, which is just under 1 hour, how can I tell them about water technologies, the importance of water technologies not only locally but globally? So I knew she also wanted me to kind of touch base in some of the technologies because of my audience, and so that's what I put together. I really encourage you to ask questions. It is such a challenge to be talking about all of this. There's a lot of going on right now in Milwaukee and globally around water technology. There's a lot going on. So let's see how much I can transfer to you within the time we will be together. And I am also available to you. I have my email right there. Please feel free to contact me to follow up on this. You are always welcome to come and visit our facilities at the Global Water Center. So, Dr. Bravo, I had the pleasure to have Dr. Bravo as my advisor, and here's a little bit of a review of the major things that are going on in my career right now. So, combined, I have 20 years of industry and academia experience. I have presented my work in multiple countries, and the two major focus of my research is development of adsorbents for removal of pollutants and also development of sensors for identification or quantification of these pollutants in water. I'm sorry, let me just get this right here. Okay, here you go. All right, so in 2014, when the EPA administrator visited the Global Water Center, she made a statement that is actually available, it's public information, where she says, when it comes to water, Milwaukee just gets it. So that's such an honor to us to get that type of a statement. There's a lot going on in Wisconsin. There's combined, there's 100+ years of industry expertise. 86% of Wisconsin borders are water. School of Freshwater Sciences is the only school in nation solely dedicated to the study of freshwater. We do have the Global Water Center, which I'm going to tell you a little more about it. So that's very exciting to be right here, literally in our backyard. So, what is the Water Technology Accelerator, or called WaTA, right? So we work with scientists, we work with business partners. The idea is to take these water technologies closer to commercialization. Water technology is a $483 billion global market, and Southeast Wisconsin owns only 5% of it. So there's plenty of room for development and increase of this market in our region. So the Global Water Center is located about 10-15 minutes driving from here. So it's very close. Global Water Center is the name of the building. So it's a business building. There are startups in the building. There are water industries, large water industries such as A.O. Smith, Badger Meters, Rexnord. Also, the Global Water Center houses the Water Council, which is the entity that connects water business, not only in Milwaukee, but nationwide and globally. Also, Global Water Center houses universities. So we have University of Wisconsin-Milwaukee on the 7th floor of the building. We have Marquette University on the 6th floor. So it's a very dynamic community. That's where everything about water comes together. Just across the street of the Global Water Center, there's something that is called the Reed Street Yards. So it's a water technology park. It is a Milwaukee investment. The idea is that that space can be receiving companies that have something novel in the field to be showcased. Right now, there's Watertronics, which is a startup. They capture water from the canal, they treat it, and they return it back to the river. There's also Zorn, which is a branch of Rexnord. They have a facility there solely dedicated for training their engineers. So it's very nice to have, for Milwaukee to have all of that right here, right? So we have, according to the Water Council, we have about 180 water-related companies in the Great Milwaukee. So when people talk about water technology, several things may come to their minds, right? And the Water Council defines water technology as solutions that address water quality and quantity for water users. Let me tell a little bit about our facility, the WaTA facility. So, by the way, that is a shared facility, so that is available to all the schools and departments at the university. So it is available to you. We do have clean room, so it is an environment. It's specifically for microfabrication and for fabrication of certain steps of filtration materials. It's an ISO class 6, so it's an environment that has limited number of particles per area, right? Researchers wear special garments when working in that room. So that's mostly for microfabrication. Microscope room is also available to all the students and faculty in our university. and to our partners as well. So other universities, industry members, this is all available to them as well. Microscope room, we focus in disinfection technologies. So we have a confocal scanning microscope, a upright microscope, and a Raman microscope. And we also have a general material sciences lab with a variety of equipment for evaluation of material properties of our sensors and adsorbents. We make sure that we can collaborate with everything, all the departments that are in the resources that we have available at the university. We collaborate with Office of Undergraduate Research, School of Freshwater Sciences, Physics, Shimadzu Center, and also AAF, and Steve is right there. We collaborate a lot with his facility. So that's the beauty of this, is really joining efforts and collaborating and bringing things together. In our facility, we have researchers specialized in certain areas. So we have researchers working with sensors, or researchers working with electrochemical or holographic sensors, material sciences, computational fluid dynamics, and mechanical pumps, aeration, and bubbles, for example. This is my group from fall 17. I want to tell you a little bit about what my lab is doing right now. My research is mostly focused in development of novel porous materials. Here are some of the students that I have assigned on those projects. I'm also working on bacteria detector sensor, biofilm, sensor for detection of biofilm in pipes, also evaluation of a device for detection of dissolved metals in water, microbial fuel cells. I have a project in collaboration with local hospital in the medical field, Odyssey project, which is a French startup, and we are helping them to evaluate the ability of their materials to reduce corrosion. I also have specific work in development of techniques for metal oxide deposition, and also for evaluation of micropores and mesopores and macropores in adsorbents. So this is what I'm doing right now. Specifically talking about sensors, and that's one of the topics that Marcia asked me to kind of touch base. I'm going to cover two of them here. Our particle and bacteria sensor. I do wanted to kind of bring to you some of something that is all the time present to us when we develop technologies, is the importance of intellectual property and the protection of intellectual property. So every time we are conceiving ideas, developing novel technologies, we think about protection of that IP, which belongs to the university. So some of the work that I will be showing here to you, you may find a little shallow, but that was on purpose. Most of our research is actually being considered by University of Research Foundation for patent. So we are not allowed to release information. It is considered public disclosure if we do so. So I will kind of show you the technology, touch base a little bit on the principles, and then just step back. I will address questions as they come. If I cannot talk too much about the technology, I will let you know, and that is the reason. So I have been working on this project since 2013 with my collaborators, Thomas Hansen from School of Freshwater Sciences and Dr. Rudy Strickler from biology. We have initially this sensor is for detection of particles, so visualization and quantification of particles, and also for detection of bacteria. So we have some progress in that field. There's a lot to be done. There's still a lot to be done. I will show you some of our progress today. As a sensor for detection of particles, one of the applications we see is evaluation of quality of dialysis bag. So that is, when talking to my collaborators at the Medical College of Wisconsin, what I learned from them is in these applications, they don't care about how much is there. They care about, is there anything there? So it's a yes-no test. Is there anything there? If yes, they cannot use the dialysis bag. If no, then of course they have to go ahead to use it safely. So this sensor is it's an optical sensor. So we use what is called holography. So images, there's a volume, a tiny volume. I believe we are working with 2 microliters right now, where the images are acquired in layers. So the number of layers and the thickness of them are determined, and we acquire images in 2D, but because they are acquired in multiple layers, so they give us a 3D information about the number of particles in that field of volume. So this is a poster that my students put together in some of these. There's a poster competition here in the College of Engineering, and this is one of the posters that they presented. There, in that picture, you see one of the old prototypes. We are probably in prototype 4 right now, so it looks different from what you can see right there. So the first time is actually acquisition of the raw image. Then there's extraction of the noise, and then image reconstruction, which is done by software, and then visualization and quantification of these particles. In this particular case that the students presented in this poster, they actually spiked the water with bacteria. So it was pure water spiked with a known amount of bacteria of a single strain. So in that particular case, we were able to quantify bacteria under these conditions. So here, one of the images we acquired when we were partnering with a startup called Stonehouse. We had our device integrated with their filtration system. So this is one of the images we acquired there. Here, I know this image is dark on purpose because that's how we see it when we acquire the images. We extract the noise, and then what remains are just these white dots. And then we have a particle tracking software where we can actually identify the particles and quantify them. I just have I will go with it again so you'll see where they are and how they were identified. So this is it's just to touch base in some of the technology that's out there. Another one, and this one in particular, we are working on patenting this idea. So this one, I tell you the purpose. I will show you a little bit of what we know so far. But at the same time, we don't release information about the technology. So right now, sensor for detection of biofilm in pipes. Let me just, my audience, just kind of bring everyone together in terms of what biofilm is, right? So we have a colony of bacteria, and they are, they live in colonization, right? We have the formation of biofilm. This is a well-known theory, right? Initial attachment. In the initial phase, there's a reversible attachment until it finally reaches irreversible attachment, maturation, and then dispersion when the biofilm starts to come off. So, why do we care about this? Well, biofilm is well known in all sorts of medicine applications, right? Not only the room where the patient is located, right, and the need for reducing the presence of biofilm. And yes, I'm talking about reducing because biofilm will grow in all sorts of surfaces, in all sorts of temperature and conditions. They may grow less or more but they will certainly grow, and that is a concern in the medical field. For odontology, right, for the bacteria, the biofilm that grows in teeth, right, or contact lenses, which I actually wear contact lenses, and that's always a concern for the users, right? So this is just a few of reasons why we care about biofilm in the medical field. And of course I have, so here this leads to 2 million cases annually in the US, costing the healthcare system over $5 billion in additional healthcare expenses. So that's by itself is a pretty good reason for us to care about presence of biofilm and quantification of biofilm. Here we have a little bit of presence of biofilm in food industry. Actually, one of my first degrees was in food industry, in food engineering. So a food engineer is a chemical engineer specialized in food industry processes. So in food industry, we care about biofilm. If you are working with food, you definitely have richness of protein and carbohydrates and moisture. So this is just the perfect recipe for formation of biofilm and growth of biofilm. We also care about presence of biofilm in the pipes. So we care about growth of biofilm in these pipes, and not only for food industries, but a variety of other types of industries. We also care about corrosion of the machinery, right? So if there is growth of biofilm, biofilm may lead to corrosion as well, right? So these are just a few of the reasons that the food industry and beverage industry is definitely one of the industries that most care about this topic. And in drinking water industry, of course. So biofilm formation inside of the pipes, first of all, it is challenging to detect the presence of the biofilm. There are some technologies out there, right? And the second thing is once the diameter of the biofilm is taken, just like the figure we have right there, so there's a lot of costs for the transportation of this water, and most importantly, health concerns, because that's the water that you will be receiving in your house, right? So lots of reasons for all of us to be interested in that. Yes, there are techniques out there for quantifying, for identifying biofilm. It would be great to have a technology that could identify and quantify the presence of biofilm in pipes if it was a non-invasive technique, right? Right now, what is done, there is coupons, right, are removed from the system, so we can quantify the amount of biofilm growing that surface. How about if we could have a non-invasive way of just scanning your pipe and knowing what is there and how much is there of biofilm? So that's what my lab is doing right now, but that I cannot tell you more. So that's just, I see some of the smiles. Oh, now I want to know what you want to say. Maybe next year when we have all the patent cleared up, then I can come and talk about the details of what you're doing. I'm actually, by the way, I'm very excited about that project. I have a student from electrical engineering from this school working in that project. I'm very proud of him. He's doing great work in that project. Now, switching a little bit to the adsorbents. So let's talk about porous material for removal of pollutants from water. So I have, it's a technology platform. So the way we do it is we work with natural material. We functionalize those materials with either nanotechnology or biotechnology in making that material specific for removal of certain pollutants from water. So one of them is technology for removal of phosphorus. We filed a patent in August of last year, so we are still waiting for, it takes about 18 months for the patent to be cleared. As soon as it's cleared, that's when we send manuscripts out. Otherwise, if you publish a manuscript before you have a patent, then you don't have a patent because it becomes public knowledge. So we hold the manuscripts until we get the patents out. So that's, I want to tell you a little bit about this. This is a very exciting project. This is a Great Lakes Protection Fund project that I have in my lab. This is a 3-year project. I have a graduate student, a Ph.D. student from industrial engineering working on this project. And also I have 3 to 4 undergraduate students working on this project. One is from industrial engineering, and I think I have 2 from environmental and 1 from conservation science working, supporting the graduate student on this project. So the goal of this project, first of all, this is in collaboration with, there are multiple parties involved. We collaborate with an engineering company based in Michigan, and they are responsible for helping us deploying this technology in the field. This technology will be deployed in 1 farm in Wisconsin, 1 farm in Michigan, and if time allows, another one in Ohio. So the idea is that the farms, they have a lot of presence of phosphorus, right? Some of the most modern farms, they have tile drains. So the storm water is collected, and they can either just discharge these in the waterways, right? But there's a lot of regulation involved on that as well. So, and we are partnering with Sweetwater, so they are helping us in the policy part. So the idea is if the farmers can reduce the amount of pollutants being discharged in the waterways, they receive encouragement, like financial encouragement. So they have costs deducted from their taxes, so it becomes very attractive to them. So what is remaining is, okay, what are the technologies out there? So this is all good for everyone, right? But there's one important thing missing in this whole story is, what is the technology out there? If that is the deal to reduce the discharge of phosphorus in waterways, what are their options as farmers, right? So we developed this technology we conceived the idea in 2014. We filed the patent in 2017. And now we are working under this Great Lakes Protection Fund grant to deploy this technology. How is this going to look like, right? So there is a picture here. The idea is the adsorbent is going to be placed inside what we call tea bag. So it's nothing else than a double geotextile layer. We place this adsorbent inside, and the water from the tile drains is collected in, so they're all combined in pipes. So before they reach the waterways, right in the middle, right there will be, the water will be passed through these bags, right, this adsorbent, and then discharged into the waterways. So this is very exciting to us. We do have now capability for fabricating these materials in up to 50 kilograms batches right in the lab. So in the beginning, when we conceived this idea and then we tested, of course we were testing 500 grams. So, but for this type of application, we need large scale fabrication. So this is very exciting to us. The way that this is going to be deployed, we are thinking about what we are calling a cooler box. That's how it looks like. Just think about a cooler box, and inside we place frames, and inside of these frames we have the adsorbents, right? So the question is how many of these tea bags we need in series to treat the water that is coming out of the tile drains. So this is the test we are performed in the lab right now. we plan to have this technology deployed in Michigan and Wisconsin this spring. So this is very exciting to us. So switching gears to the adsorbent for removal of persistent organic pollutants. So the difficulty, it's very exciting to me to be working with persistent organic pollutants. The challenge here is that most of the persistent organic pollutants, they either don't have any sort of policy, any sort of enforcement from EPA, or they have it, but it's as a form of recommendation. It's not enforced. Why is that? So when we are talking about, for example, metformin is the active pollutant, the active ingredient of medication for diabetics people, right? So that's the active ingredient. So when you take your medication at home, right, you ingest that medication, but that medication is discharged, goes back to the waterways through urine, right? So it goes back to the waterways. That sounds great. Okay, now we have to treat it, right? It turns out that one of our researchers at the School of Freshwater Sciences, Dr. Rebecca Klopper, she did a study that she collected samples in different steps of the drinking water treatment plant and wastewater treatment plant. And turns out that for this, and I think she probably has, this is already published in probably 3 years ago or 4 years ago. So this is well known in the literature. So it turns out that she was able to show that metformin is one of the pharmaceuticals that is not removed from any of the unit operations in the treatment process, right? So what does that mean to us? When I go to the sink and I grab a cup of water, what does that mean to me? What does that mean to our children? We don't know. However, as a toxicologist, she did laboratory work and she published. And in one of her publications, she showed that aquatic species, so again, we don't know what happens or what are the impacts to humans, and that is one big reason that EPA cannot have a regulation, right? But as a toxicologist, what she did, she studied the impact of this pharmaceutical in aquatic species. And she identified female features in male specimens when exposed to certain levels of this pharmaceutical. So this is where things start, that's when we start understanding a little bit more. It doesn't mean the same happens to humans. All that means is this is a toxicological study with aquatic species, right? Studies with humans is way more complicated, and perhaps as part of my collaboration with the local hospital, we are also focused on glyphosate. So this is just the tip of the iceberg, right? There's a lot to be learned, but until we as a community, the academic community, not only me, until we as a community gather information and gather documentation, there's no way to build policies without data. And that's what the researchers are trying to do. So my student did, she developed an adsorbent in the lab. We specialized that adsorbent for different types of organic compounds, cationic, anionic, and non-ionic compounds. She also tested, and the great thing about this adsorbent is that it had high % removal for this variety of model compounds. Then she went on and she tested these compounds, this adsorbent, with 2 persistent organic pollutants. In her case, let me see if I can't really see here. In her case, I think she tested with We tested with metformin, we tested with atrazine, and oh yes, she tested with PFOAs, and she tested with another one. We tested with PCBs, and there's another one that I can't remember right now. But PFOAs and PCBs we also tested. So we obtained great laboratory work. However, remember, laboratory work is a controlled environment where there's clean water spiked with a known amount of these compounds. These experiments were performed one at a time, so there was no combination of these compounds when experiments were performed. That means we have not tested for competition yet. There's also another adsorbent that my graduate student in the Department of Material Sciences is working on. We developed an adsorbent for removal of heavy metals. So his mostly focuses on the removal of lead and mercury from water. So you certainly remember about the Flint case in 2016, right? And at that time, the major concern was the impact in children, right? Because it may cause development delays and a lot of a series of concerns with their little bodies. So that's when this came to the news in 2016. The problem is, I believe also in 2016 or 17, of course then there were many researchers publishing, right? Because it became the topic on the news. And then that was one case that came to the news, right? So everyone knows about the Flint case. But then other studies have shown that actually there are concerns with lead across the 50 states. It's not a Michigan problem. It's all over, right? So that is the concern right now. So there are old infrastructures, right? So that is one of the situation or one of the sources. But the other thing is the EPA considers safe 15 ppb or less of lead in drinking water. In fact, there's no safety when you are ingesting lead, right? What there is a number that is at least is safer to the consumer, right? So the idea is, can we be challenged and develop technologies that can reduce these levels of drinking of lead in drinking water, right? What is that going to look like? So there are many. I'm going through a few slides and just jumping a little bit. There are many technologies or developments right out there, right? It's not only adsorbents. There are other technologies that can potentially be explored and be used for the removal of heavy metals. In the particular case of my student, he mostly focused on ion exchange and adsorption, right? There are other mechanisms. So he also tested how the behavior of this adsorbent when exposed to different pHs, right? We do care about that. Depending on the incoming water, the performance of the material may change. He also tested temperature. From his study, I believe he tested from 4 to 50 degrees Celsius. He didn't see much impact on within that range of temperature. And he also tested the RPM. So what we were looking at here is more impact of the laminar flow or turbulent flow of this water when passing through the adsorbent. So he did explore that in terms of efficiency of the filter. So this is just a little bit to touch base on some of the technologies that my lab has been developing. We have also been working with adsorbents for removal of ammonia from water, Legionella from water. There was a company based in Illinois. They work with healthcare, and they came to us and said, hey, I know you are working on an adsorbent for removal of E. coli. which for me was just a model organism. And they said, would you or could you be interested on working with adsorbent for removal of specifically Legionella, right? So we start working on that. And my student, actually, the engineering poster competition is coming very soon, and she is getting ready to gather her data and be presenting I believe it's going to be in April, the competition. So this is very exciting to me. There's a lot I wanted to talk to you. I wanted to touch base a little bit in water hubs worldwide, but let me gather some questions right now, and then if time allows, then I talk a little bit more, because I can talk more than I'm supposed to. So let you talk first, and then if time allows, I will give you a little more information.
Thinking about, well, phosphorus in agricultural situations and mercury that reaches waterways in rainwater. There's a lot of non-point inputs for those two things. Are there applications for using the things you're developing with non-point sources?
Absolutely, yes. Thank you for the question. So what we think is literally the deployment of these tea bags in hotspots in the city. Let's say you identify hotspots with lead contamination, so you can literally have in the manholes these tea bags, so the water, as it before reaching the pipes, it goes through the bag first. So that is one possibility. And the technology is very flexible, the way it can be deployed. It can also be used literally in a filtration system in drinking water treatment plant or wastewater treatment plant, in situ remediation. There's one work we did in, actually with Professor Bravo was one of the co-PIs in that project. We collected sediments from the bottom of the lake in Green Bay, and in the lab we quantified concentrations of mercury and PCBs, and now my students are testing laboratory scale, but they're testing the remediation, using this adsorbent as possible remediation technologies for the sediments. And just yesterday or the day before, I was talking to an engineering company locally, and his major concern was with presence of PFOAs in soils and contamination of groundwater. So now we are starting experiments in the lab using similar type of technology, but the right adsorbent for pharmaceuticals, right? So, but we are exploring now these adsorbents for in situ remediation. So I'm excited. This is ongoing research in the lab. I don't have results yet, but I can't wait to get these results. More questions? Professor Bravo.
For your dissertation, you worked on the quantification of the attachment of bacteria to sediments using a procedure that use a certain type of bacteria and shining a laser sheet, taking images, and then image processing. So for these sensors for the detection of bacteria, what are the main differences between what you did for your dissertation and this other sensor that you developed?
Well, this other sensor, in collaboration with Rudy Strickler and Thomas Hansen, is optically based. So is the other one, but the difference is we use holography. So we acquire images in different layers, and then they are raw images. We have to reconstruct them and then make them become more, if I were to say, the images become more clear, right? And then we can identify the cells and quantify them. So they have different principles, right? And you are correct that we do use laser in both cases, but the one that we worked in the lab, we didn't work with holography.
talking about biofilms.
Mhm.
Are you working on surface treatment so that the biofilms don't initially settle on the surfaces as opposed to cleaning them?
I love the question, and I love the way it happened in the lab. It didn't happen on purpose. So the answer is yes, but I wanted to take a minute to tell you about that. So when my student, so she's a microbiology student, right? So she was really interested in biology. But at the same time, I had to combine her interests with my interests. So she wanted to work on biofilm. I'm really interested in the water technology and some use for water industries. So her project was to choose 2 adsorbents in the lab, seed them with bacteria, let bacteria grow. So she programmed the confocal microscope to acquire images every hour for 45 hours. So she had a natural material and she had our engineered material. All I wanted for that time as a student for her, I wanted her to gain the skills and I wanted her to quantify biofilm, which was her interest. Turns out that her research was even way better than I could possibly think. So her research showed that our engineered material had the property to reduce the rate of biofilm growth on the surface of the porous material. And that is not, when we conceived that idea, we were not thinking about that. We were thinking about removal of organic pollutants from water. And then it turned out that the way we engineered the material also had anti-biofilm properties. So that was pretty good bonus for us out of that research. So the answer is yes, but it was not intentional in the beginning.
So thinking about the project with the phosphorus and the drain tiles, that's one situation where the pollution is actually a resource out of place, that the farmers deliberately want phosphorus to improve their soil, but then they don't want it to enter the waterways. I guess would that technology provide a way to measure the phosphorus so that farmers could be alerted if they're over applying, and also is there a way to recapture the phosphorus for applying to the soil?
Thank you for the 2 questions. So let me start with the second part. So when we adsorb phosphorus in this media, the idea, and I failed to mention, thank you for the question, the idea is to adsorb phosphorus and actually recycle the media, right? So you can reuse it, but also recover the nutrient, and very importantly, have it used back by the farmers in their place. So that's one thing. Another thing about the sensor, right? I don't have any project in the lab right now working on detection of phosphorus, but that is absolutely something we could do. It would be wonderful if they could have a way of monitoring the farm, right? Instead of just understanding the consequence of their doing, if they could have information in nearly real time of the impact of their procedures. So thank you for the question. And that's the beauty when we receive lots of visitors in our facility. And I love interaction with the public, especially because this type of question, the things we already do, but things that with fresh eyes, people can give us the idea of what they believe could be another possibility of application of the technology, or another field that we could be focusing on. So that's absolutely amazing. Thank you. More questions? All right, I know I have a 12:50.
Maybe one
Yes.
quick question. You mentioned that you have computational fluid dynamics capabilities.
Not in my lab, yes, in the center.
Can you elaborate a little bit what are those capabilities?
Yeah, absolutely. This is mostly Dr. Rio Amano's lab. So what he does in the lab, I will tell you about one of the projects he has, which is partially funded by Cadence, which is a startup, and partially funded by Rexnord. So what he develops in the lab is a hydro turbine. And his students, they design their own turbines, and they literally they 3D print it in the lab. So they can, they have a pilot plant in the lab. So they can test the different designs, different turbines, gather information from the system, use computational fluid dynamics to model the system, and eventually what they really want is scale that system up and have it deployed in the Cadence place, right? So it's in another city. So that's the idea, learn from the system and then be able to scale up. And I think, is it 12:50 right now? Is it? That's it, right? Okay. I would like to thank you so much for your time today. I had so much fun preparing these slides and so much fun presenting it to you. So thank you for your attention today. Okay, let's keep in touch.